The gynoecium is actually the female reproductive part of the flower, or in a flowering plant. Now the gynoecium, it is or it is also called the pistil, is actually the part which is the central most unit of the entire flower, and it majorly comprises of the stigma at the top along, or the length of the style can vary, so it's a long thread-like structure which is connecting the stigma with that of the ovary at the bottom, and it is in the ovary where we will find that the ovule or the egg cells are actually present there. Well, in reality, the ovule is actually a large structure inside which there will be an embryo sac, which will contain the egg cell. So, talking about the structure of the entire pistil or the entire gynoecium, the topmost layer or the stigma is actually a very important part, because it is the place where the pollen comes and adhere to the flower. So means the female part of the flower. Now this is actually the structure which is a kind of a receptacle. It accepts the pollen, and then the pollen will start germinating from there, resulting in formation of the pollen tube, which will gradually then fertilize the ovule at the ovary. Now, the talking about the structure, you can see this is actually a scanning electron micrographic image of the stigma, and you can see the left hand side one is actually the image of the this like China rose plant that we normally see all around home everywhere. So it's the stigma of the China rose. We can see this structure. It looks like a hair-like structure right here, but in reality, those are actually the receptacles that are extending outward. Some pollens can be seen adhered to the surface of it, but I'll ask you to see this image rather, where we can see that the pollens are actually adhered to this particular stigma. It's a different flower. It's stigma. The pollens are adhered to it, and you can see that the pollen tubes, which are these like tube-like structures coming out of the pollen, you can see that it's going and penetrating inside the stigma. So that is actually the pollen tube, and on the other side, on these two pollens, you can see that the pollen tube is actually forming itself. So it is right there where the interaction between these two actually takes place, and the resultant pollen tube is actually forming right there. Now, talking about the interior part, it is actually this structure, which is the cross section of the ovule region, of the ovary rather, is where we can see actually the main reproductive cells are present. So you can see this structure that is present here. This empty chamber are actually the locule region where the ovules are present, and ovules are those darkly stained central bodies that you can see in this clear halo. So this is the area, this is the place where the each and every of those ovules are present, and the ovule is actually the one which will be fertilized, then will transform into the means the seed later on. This is a longitudinal section of the same means ovary. You can see there means the entire part has been labeled here properly. So we can see right here is actually the entire flower. This is the cross section of the entire flower. That's why you can see the anthers are also, it can be seen on the side. So this is the central body where you find this is the ovary region, of which there are numerous styles and stigmas like originating out of it. So from there we find like here is not, it's not having a single, like each and every of the ovary or each and every of those ovule ovary actually is what is having its means the stigma or the style, the entire pistil, they are all being separate, and they are all originating from the same spot we can see here. And the ovary is carrying the ovule inside, which you can see there are two ovules present per ovary right there. So this is a cross section of a flower showing the entire structure, how it appears, how it looks like, everything. Now, talking about the ovule, the reproductive structure. So it is actually a structure where we will find that it actually contains this entire ovule. It remains inside a structure which is called the locules, and the boundary of the locules, or the cushion in which these locules are actually present, or these ovules are actually present rather, is called the placenta. So it is a placenta which is the cushion parenchyma. It's made up of majorly parenchyma cells, and they are the cushion in which this each and every of these ovules are present. So an ovule will be surrounded by a placenta, definitely. Now, the ovule, it's actually connected with the placenta with the help of this stalk-like structure labeled as the funiculus. Now, funiculus is a like it's simply as like a stalks with which the ovule remains adhered to and remains suspended even. So this is the one which is all connected right there. And you have to keep in mind is actually the funiculus. It connects with this entire ovule body, and the place where the adherence actually happens, or the place where these two unit they adhere together, it produces this little a kind of a groove which is called the hilum. So hilum is the point of attachment of the funiculus with the main ovule. Now, the ovule, it contains a large mass of parenchymatous cells called the nucellus, and from there, finally, later on, it actually the main embryo sac actually it develops out from there. The this is actually the place where the you can say the embryonic development will take place later on. So the embryo sac is located inside the nucellus, and it will be multicellular structure, definitely it will be discussed in the next slide. Now, the outermost multicellular covering of the ovule is what is known as the integument. You have to keep in mind these integuments are actually the one which will form the seed coat when this embryo sac or this egg cell they actually get fertilized, and then this entire embryo sac is transforming into a seed. At that particular moment, it is these integuments which will transform into the two layers of seed coat called the testa and the tegmen. They are formed from the integuments. Now, they have a narrow pore. These integuments actually are not entirely covering this embryo sac, but rather there is a small pore right there called the micropyle. And usually it's the center from through which, or the side through which, you can see that the germinating tube actually enter inside the inside this ovule, you can say. The extreme opposite corner of this means the other extremities of this micropyle is where we'll find that there is a basal, like it's actually like a base like structure to which the entire ovule is suspended, we can say, is what is known as the chalaza. And rarely through that area that an entry of a pollen tube can be noticed. Usually the pollen tube enters through the micropyle region, but in a rare case also we can find that it may contain this, it may enter through this particular side. This is an diagrammatic representation of the mature embryo sac, where we will find that the egg cell is actually located in the corner. Now let me tell you one thing: the egg cell, the direction of the egg cell is always towards the micropyle. It always remains towards the micropyle ends. That means the opposite end is actually the chalaza. So this micropylar end, we find that this egg cell, it has these two cells, larger cells which are connected with it, which are called the synergids. And synergids is just like a nurse cell for the egg. Means it is the one which nurtures and which helps in maintenance the sustenance of this particular cell, and later on is also going to be the site through which the male gamete will enter inside this ovule. Now, the polar nuclei, which is present at the very center of it, are actually two in number, and these two polar nuclei are inside this large structure called the central cell, which is the major portion of the entire structure. And this central cell body is the one which will later on transform into this endosperm. We know it's one of the part of this entire seed, and this endosperm is actually developed. It develops from the central cell, and the egg cell after fertilization becomes the main zygote, which will transform into the main embryo of the plant. Antipodal cells, they are just like three cells which are present towards the opposite end to that of the synergies and the egg, and it's actually just like the counter copy of this like egg cell, but they have no function at all. Means they don't take part in the process of entire process of reproduction is just present right there, because in the mitotic division the meiotic division, post meiotic division, there is a mitotic division also that takes place, which actually leads to formation of these antipodal cells, and this remain right there for the entirety. Now keep in mind, in the entire structure you are seeing like there are 8 nucleus, and all 8 nucleus are actually completely haploid in nature, every single one of them. But there is a thing that these 8 nuclei can be found present inside 7 cells altogether. See the synergids being 2 cells, one egg cell, 3 antipodal, and then you have the entire central cell carrying 2 nuclei. That's why there are total 7 cells that will be seen, but there is only like there are 8 nucleus that are present there, because 2 nuclei are present inside the central cell itself. So that becomes the structure of the mature embryo sac. Lastly, depending on the like way, the relative position of the entire of the funiculars, the chalaza, and the micropyle of that of the ovule can be designated into these 3 different categories. This actually rather is shown here as 6, but 5 to 6 categories that we can actually distinguish them. So first one will be the atropos, or the atropos sometimes they are also called, is the simplest and the primitive type of the ovule that will be seen, where the micropyle, the chalaza, and the funiculus all lie in one single vertical plane, or the vertical plane we can say, and they are majorly seen in the case of the gymnosperms. Means the gymnosperm type of plant, they usually show this particular variety of arrangement. But not only that, we can also find this in some of the citrus family also, citrus fruit family. Anatropos, you have to keep in mind, is the most common type of ovules that actually exist. Like more than 80% of the angiosperm family that we know, it majorly comprises of this particular type of like arrangement, and this is actually also called the inverted arrangement, because we'll find that the micropyle and means the micropyle comes near the funiculus, and the chalaza is like in 180-degree opposite to that. So it, in a way, it is like the entire ovule has made a 180-degree turn. Means with the case of the atropos, it is like it's like 180-degree turn of the atropos system is what we are seeing here. So that's why this structure is called the inverted type of ovule. Next is amphitropous. In this type, the body of the ovule is as well as as well as like the embryo sac is also curved like that of a horseshoe like of pattern. And like this particular type, it is present in many of the plants. Like for example, I don't have the picture of it. It's like the butamus. I should have added a picture of it. So butamus, like families of plant, they actually have this particular type of system. Now, next is actually Hemi and Atropos, and it is somewhat in between, we can say, the Atropos and the Anatropos type. Means instead of a 180-degree turn, it makes a 90-degree turn instead. And thus, we'll find them to be present in like perpendicular to that of the funiculus. Means the micropyle and the chalaza lie in a perpendicular plane to that of the funiculus plane. So that's why they are called this Hemi and Atropos type of arrangement. The last one is Cercinotropus, and Cercinotropus is actually the type which we find in cactus family mostly, and you can say it's like the funiculus actually increases in length. The anatrop, like it's simply like that of the atropos type, but it is the entire funiculus, it becomes so long that it entirely covers this entire structure and then makes a like full turn. Or you can even like consider this to be like the atropos type of condition, which has made even like due to the funiculus, it become even like atropos type of arrangement. It's an entire coiled structure. The diagrammatic representation is already shown here, you can see it. So it's majorly seen in the cactus family, where we find this particular type of feature of ovule can be it can be noticed there. So those are the different types of arrangement of the ovules, and this is how the entire ovules are arranged. Next video, next class will be discussed I'll be discussing about the process of the megasporogenesis, or the, like the process of the like formation, okay, of this embryo sac and all.